Archana Sharma Foundation of Calcutta, Professor Amita Pal, to say a few words and proceed with today's proceedings.
Thank you, Momita. Good afternoon. I hope that your family members, your colleagues and friends all are doing well during this unprecedented, unsettled time. The period caught between the threats of the global pandemic, civil unrest, and economic crisis. I am sure that many of us never felt so helpless, psychedelic before, and at the same time, we need hope, courage, and optimistic attitude more than ever before. It is my pleasant duty to say a few words about the Archana Sharma Foundation, Calcutta, the today's host, which is a registered society under the West Bengal Society Registration Act 1961 and was founded by Professor Arun Kumar Sharma in the year 2009. The society is functioning from the Department of Botany, University of Calcutta, and is a non-profitable organization. The foundation strives to keep the legacy of two stalwarts of Indian scientific community, Padma Bhushan Professor Arun Kumar Sharma and Padma Bhushan Professor Mrs. Archana Sharma, whose achievements need no introduction. For the past 11 years, our foundation publishes the journal The Nucleus with Springer Nature as a co-publisher, and also organizes seminars and symposia either singly or in collaboration with the Department of Botany or other scientific bodies, scientific societies, both nationally and internationally. Such activities are within the defined objective, defined scope of the foundation. Additionally, for the last 3 years, since our beloved sir, Professor Arun Kumar Sharma, left us unaided, alone, we started annual memorial oration to pay our tribute to Didi and Sir. Unlike this year, usually we assembled in the Archana Sharma Memorial Hall of the Department of Botany, University of Calcutta, for such oration. This year, due to the pandemic situation, we have to organize virtually through Zoom as an alternative arrangement. Today, we have with us Professor Dhruvadyoti Chattopadhyay, a well-wisher of the foundation, to enlighten us about the most contemporary and opportune COVID-19 and its epigenetic consequences. With these remarks, I welcome you all again in today's webinar. I now call upon Professor Rita Kundu, Secretary, Archana Sharma Foundation of Calcutta, who will introduce today's speaker, Professor Dhruvadyoti Chattopadhyay. Rita, please.
Thank you, Didi. Though Professor Dhruvadyoti Chattopadhyay needs no introduction, but it is customary to introduce him to my audiences. Professor Dhruvadyoti Chattopadhyay is currently the Honorable Vice Chancellor of Sister Nivedita University, Kolkata. He served as the founder Vice Chancellor of Amity University from 2015 to 2019. Professor Chattopadhyay acted as Pro-Vice Chancellor Academy from 2008 to 2015 and as a Dean, Faculty of Science at the Calcutta University from 2003 to 2007. He was also the Director of the Center for Research in Nanoscience and Nanotechnology at the Calcutta University. Professor Chattopadhyay started his career as a lecturer in the Department of Chemistry at Jogomaya Devi College in 1981, after which he joined as a lecturer in the Department of Biochemistry, University of Calcutta in 1983. In 1997, he became the Guha Professor at Dr. B.C. Guha Center for Genetic Engineering and Biotechnology under the Department of Biochemistry, University of Calcutta. He graduated from the Presidency College, Kolkata in 1973 with a major in chemistry. Professor Chattopadhyay did his master's in biochemistry in 1975 and completed his PhD in 1984 from Department of Biochemistry, Bose Institute, Calcutta. Professor Chattopadhyay has a research experience of more than 35 years. During his research career, he had been to Oration Institute of Molecular Biology in New Jersey, USA, as a visiting postdoctoral fellow, as visiting project scientist to the Cleveland Clinic Foundation, Cleveland, Ohio, USA, visiting scientist to Stowers Research Institute, Kansas City, to the University of Texas, to Nihon University and Hosei University. He is also the recipient of numerous awards, accolades, and distinctions, which include the Young Scientist Award in 1989, member of Go Research Conference in 1992, Professor Umakant Sinha Memorial Award in 1992, Fellow of the National Academy of Science in 1998. He received the prestigious UNESCO IUMS MIRCNS SGM Fellowship to carry out research under the Department of Biochemistry at the University of Texas. He was a fellow of the Indian Academy of Science, fellow of West Bengal Academy of Science and Technology, to name a few. Dr. Chattopadhyay has lifetime membership in various organizations and scientific associations like Indian Science Congress Association, member of Asiatic Society, member of Biotechnology Society, member of Virology Society, and so on. Above all these, Professor Chattopadhyay is a wonderful teacher, a scientist and academician par excellence. During his long teaching experience, he has supervised a lot many students for their PhD thesis, and he has around 130 research papers and 6 book chapters to his credit till date. Above all, he is a beautiful person, and we all still depend on him for so many, for his energetic and enthusiastic uh, help to all of us. Now over to Momita.
So obviously now we will move on to the oration lecture that we are waiting for, all of us are waiting for so eagerly, as Professor Chobe also mentioned. We want to really listen to what our teacher and Professor Dhrubajyoti Chattopadhyay, his take on what is happening around the world now and what are the consequences. We will just take a minute to set up the presentation. I'll ask the audience, I'll tell the audience that you can ask questions. We will request Sir to take a few questions after his lecture. So you can type your questions in the chat box. And after Sir has finished his lecture, we will impose upon him and to take a few questions and assuage our queries. So though it's not a custom of any oration lecture, but I'm sure Sir will not mind there. So can I ask Mr. Mondal to set up the PowerPoint presentation? Over to you, sir.
Yeah. Thank you, Momita. Let me first take the blessings of Sir and Didi, Professor A.K. Sharma and Professor Archana Sharma. And I really want to thank the organization, Archana Sharma Foundation of Calcutta, for giving me this opportunity to deliver the third oration lecture in the joint names of late Professor Archana Sharma and Professor A.K. Sharma. I'd like to thank Dr. Amita Pal, the president, Dr. Rita Kundu, the secretary, and definitely it's very coincidental that this year I am going to discuss with you about the virology, and your head of the department right now is one of the virologists, Binay Chobe. So I also like to thank my friend Anita Mukherjee and all my colleagues who are present today and researchers and students who are present today in my lecture. Now, when actually Anita asked me to deliver a lecture in this third oration, I was thinking about what should I discuss. And then it reminds me of something. You know that my association with Didi is become very close almost 20 years back, when in her room, she actually given me a huge amount of chocolates on 24th December 2000. And that's a very interesting thing, actually. So Professor I.B. Chatterjee and myself actually gone to Professor Sharma, and Professor Sharma then actually taken us to the room of Madam Didi. So that's when I started receiving the chocolates, and then I received chocolates almost every Saturday from her. And while discussing with Professor Sharma, because she is actually working in the area of a large number of, say, cytogenetics, cytology, and all these things. And also, was pretty interested in the regular epigenetics. And I had an interesting discussion with her in the area of epigenetics with her, because at that particular period of time, almost 15 years back, one of the very interesting paper came out, which shows that the patterning of the characteristics of population, which determined by the histone modifications. That's one of the very interesting paper came out at that time. So I thought, why not I discuss, though there are very little amount of work been done in the area of epigenetic implications of COVID-19, but I just like to share with you. First, I kept my discussions, my seminar in three different parts. First is the clinical features of the, yes, fine. Next slide. Clinical features of the infection. Now, already all of you knew about that there are three phases of infection. And in these three phases of infection, what we generally find that the viral response phase, that is the early stage of infection, where from the recovery rate is very high. Then it goes to the pulmonary phase. And in this phase is also actually been divided into two. And we have the symptoms started like shortness of breath, hypoxia, as well as abnormal chest. Imaging could be shown at this particular time period. And this we call as a pulmonary phase. Even this phase, if a person enters, it's not very difficult to get that particular person back. Then the last stage, that is a stage 3, which is a hyperinflammation phase, which starts from the second stage of stage 2, the host inflammatory response phase, where we have these different shocks, cardiac failure, and the elevated inflammatory markers being identified. And we call this phase as a cytokine storm. And all the complications started in this stage 3. At the very beginning, normally we have a lot of problem if a person goes into the stage 3 and proper medication, proper protocols and other things are not being developed at that time. Though we are pretty good right now, so if somebody goes into that phase, it's becoming very, very critical. In the next slide, I just want to give you, all of you know about it in a shortly, that COVID-19, how does it affect us? Now, you know that in a healthy individual, always you can see that if you look at the alveoli, and the alveoli is very healthy, and you have the alveolus is surrounded by the red blood cell, capillary wall, and there are alveolar cells, and there are a lot of surfactant on the surface of this. And there are two different types of alveolar cell, type 1 and type 2. When it gets infected, and moderate infection, because of this particular receptor through which it goes and goes into this lungs and then to the alveoli, then we can see that starts particle formation. You can see number of things which are coming up inside the alveoli. And in the moderate infection, you can see it's becoming filled up with fluids. The fluid started accumulates, and thereby you have the cytokines, you have the vasodilation, you have the reduced surfactant, and all these things are actually in the phase 2. And then it goes to the phase 3, which is severe, when you can see almost it is filled up with the fluid, which is a protein-rich fluid. And the fluid-filled interstitium, there are loss of surfactant, neutrophils accumulation, protein and cellular debris are accumulated in the alveoli, and that completely hinders the gas exchange. Next slide. And over here, I can tell you that in the type 1 cells are thin enough that the oxygen passes right through, but the type 2 cells secrete surfactant, and that actually from collapsing it during the viral infection, you can see that the RNA from the virus, it goes into that primarily on the type 2 alveolar cells. And then we could find that there is impairment of the gas exchange. And here we could start looking at the different types of immune responses, and there are a lot of cytokine release, neutrophils are recruited, you have acute respiratory distress syndrome, the systematic inflammatory response syndrome, the SIRS, as well as it started the septic shock. And that is how we start getting the organs affected in COVID-19. In this slide, I just showing you the invaders impact. So in serious SARS-CoV-2 lands in the lungs and can do deep damage there. So in the lungs, it damage, but it also damages heart and blood vessels. And in the heart and blood vessels also, it attacks, cardiac inflammation, all these things. Then it goes to the brain. It can go to the brain, and you can see recently large number of this neuro shock of these patients are actually coming. It also have the eyes, conjunctivitis and inflammation takes place. Nose is the first through which it starts going out. The virus may move up the nose nerve endings and damage the cells, the liver, the kidney, the intestine. So you can see almost the large number of organs gets affected with COVID-19 infection. Now, if we, next slide, I just wanted to show you, next slide, the putative association between the severe COVID-19 and the indicated pre-existing chronic diseases. So say for example, if you have the CVD or the hypertension, you can see that there are acute cardiac injury. This blue line indicates the association between the pre-existing chronic disease and the COVID-19 severity. The red line indicates the organ injuries. And then you can see the expression of ACE2 in the indicated organs is also indicated by the brown line. Now, you can see that acute kidney injury takes place if you have a chronic kidney disease, whereby a huge amount of creatinine goes up, you have burn, you have the urine output. The acute liver injury is somewhat, we could see, it is not associated with any pre-existing chronic disease. The diabetes, we know that it definitely has got a huge association, but we really are very little information about that, how actually in effect the organ injuries observed in pancreas. The other organ injuries which are actually been observed is a brain, testis, eye, etc., which has also got this ACE2 receptors on it. In the next slide, I start my discussion. So this is just a background to actually make this whole thing set up. Now, discovery of coronavirus, the first person to isolate this is 229E strain. Before that, there is one strain been identified, but it's actually been lost. But Dorothy Hamry, the virologist and infectious disease researcher at the University of Chicago, Department of Medicine in 1966. And then Jules Almeida, working at Britain's Common Cold Research Unit, Salisbury, produced the first image of the virus. You can see that first image, the spike proteins which are coming out, just like the things which are coming out. And this actually, we can find it out, the viral envelope, it consists of a, next slide. The viral envelope, just if you look at, just looks like a beautiful flower, and it consists of a lipid bilayer, membrane protein, almeida protein, spike proteins are anchored, and a subset of coronaviruses, specifically the members of the beta coronavirus subgroup A, So there are different types of coronaviruses, but the one which is affecting us is a beta coronavirus subgroup A. Also have a shorter spike-like surface protein called hemagglutinin esterase, HE. So we have the spike protein, M protein, IM protein, which are all related with in some way or other for the virulence. In the next slide, I just wanted to show you a schematic, next slide, schematic representation of the SARS-CoV-2 structure. So this is actually the structure, and you can see the organization. There are ORF1A, ORF1B. So these are the different proteins which are being coded by ORF1A and ORF1B. And we have also a papain-like protease, PL-PRO, and 3CL-PRO is a 3CL protease. Both these proteases are very, very important. We have the RNA-dependent polymerase, helicase, endoribonuclease, all these things are there. And when we actually go to the next slide, it is very interesting that right after this virus been almost looked as a pandemic throughout the world, 2020 March, 11th March actually WHO announced it as a pandemic. So within 1 month, actually the electron micrographic structure of the and also the cryo-electron micrographic structures are being identified and being published. Now the schematic portion of the figure shows the structural E and M structural proteins of the membrane, protruding trimeric structural protein, and all these things are why important that the S protein, if you look at the spike protein, this spike protein with the receptor binding domain RBD, you can see the RBD is there, and with the domain that binds the AC2 receptor. And also you have the structural protein A that binds this ribonucleic acid and the RNA genome. In the next slide, when we looked at this genome organization of this human coronavirus, the genome is approximately 27 to 34 kilobase, the largest among the known RNA viruses. And we have the replicase ORF1A and ORF1B occupying about 2/3 of the genome and is directly translated from the genomic RNA. And the structural and accessory genes, however, are translated from the subgenomic RNA or the sgRNAs during genome replication. And there are almost 7 strains so far being identified. And during the transcription of coronavirus, this subgenomic messenger RNA which are being produced is used to synthesize the polyproteins and many structural proteins via ribosome frame shifting during translation. In the next slide, I just wanted to show you that these strains, the timeline for the emergence of this human coronavirus. And you can see this human coronavirus NL63 is there, existed long time back, 560 to 820 years ago. Then HCOV229E, the one which has been Dorothy Hamre been identified, and it almost diverged 200 years ago. And 120 years ago, we had the image roughly 100 this human coronavirus OC43. Then we had the human coronavirus HKU1 with that extant lineages traced back to 1950s. And the SARS-CoV, that is the one which starts first the worldwide attention of this infection, being actually from bat which has been transmitted. The SARS epidemic has been recorded in 2002. Then the MERS-CoV actually, which is actually Duronomicia capensis beta coronavirus, traced back to 2006 in the Middle East. And then we have this, our latest one. Just quickly going through this, just pointing out one of the most important area of this life cycle of SARS-CoV-2. As I mentioned to you, it uses the receptor AC2 for entering the host cell, and it binds to the AC2 with higher affinity than other coronaviruses, SARS-CoV-2. And that's why you can see, as mentioned by Billy Chobe, the huge number of infections we could identify. And this is due to its inactivation by a host enzyme called furin. The spike protein of SARS-CoV-2 is primed by another protein called TMPRSS2. This is an interesting thing. You can go to the next slide. Next slide. This next slide, I just explained to you that the spike protein is primed by TMPRSS2. Now, if we just make a comparison between the 2002 virus versus the recent virus, both the RNA genomes are almost 80% identical. And you can see that SARS-CoV-2, SARS-CoV was more aggressive than SARS-CoV-2. But that 2002 virus, it is not really so infectious so that you can go to a huge number. So you can see that if the virus is more aggressive, it has got a limitation for the virus itself. But SARS-CoV-2 spread faster, sometimes with hidden symptoms, allowing each infected person to infect several others. And that's why you can see the R0 value for SARS-CoV is 0.4, whereas SARS-CoV-2, it is almost 1.4 to 2.5. And there are so many different things which you can generally find out in both the cases. But the last point is very interesting. The case fatality rate of SARS-CoV is 9.6%, whereas the SARS-CoV-2, it is 2.3%. And in our country, we could see that it came down to almost 1.57%. Next slide. And it can spread faster because an activation sequence S1, S2 cleavage site, which is similar to those actually observed in case of influenza virus. In today's newspaper, you can see there is one particular medicine which is very effective in case of influenza virus, was found to be very effective also. The US government actually announced that is a molnupiravir, which actually can really a good one which can act on it. But there is no relation between this influenza and this SARS-CoV-2 virus. But this particular previous site similarities are very interesting to find it out. And this S1, S2 activation sequence of the SARS-CoV-2 spike protein is cleaved by the cellular protease furin, and that this cleavage event is essential for the infection. So if furin can be inhibited, then you can see the infection could not happen. It is also important for the fusion of infected cells with non-infected cells, which might allow the virus to spread in the body without leaving the host cell. So both this particular protease, S1, S2, the furin, and the TMPRSS2, S2 are important. And these are actually people are working with so that we can develop some medicine against it. In the next slide, I can show you that the SARS-CoV-2 infection can spread faster, the one I'm mentioning to you. In already infected cell, the enzyme furin, which cuts the spike protein at the S1, S2 site. The spike protein then mediates viral attachment to a new host cell. In order to efficiently enter the cell, the spike protein still needs to be activated by the enzyme TMPRSS2. And this activation by TMPRSS2 is only possible if the spike protein has previously been cleaved by furin. So this is actually what we can find, that furin is present in various human organs such as liver, lung, small intestine. And the fact that this enzyme resides in all of these human tissues means that the virus can potentially attack several organs at once. So this is how actually I can show you the pre-activation followed by the virus attachment, followed by the activation S2, the spike protein, and then the cell entry. So this is how actually what we can find already infected cells, the new target cell you can see, and this is the reason why the infection can spread faster. So now the chronology of infection, if we look at in the next slide, and you can see that this chronology is very related to the infect cells expressing this SCE2 and TMPRSS2. The active replication and release of the virus cause the host cell to undergo pyroptosis and release damage-associated molecular patterns, including ATP, nucleic acid, and ASC oligomers. And these are recognized by neighboring epithelial cells, endothelial cells, and alveolar macrophages. triggering the generation of pro-inflammatory cytokines and chemokines, including IL-6, IP-10, macrophage, inflammatory protein 1-alpha, 1-beta, and MCP1. These proteins attract the monocytes, macrophages, and T cells to the site of infection, promoting further inflammation with the addition of interferon gamma produced by T cells and establishing a pro-inflammatory feedback loop. So this is how actually the SARS-CoV-2 infection proceeds. And then you can see that in a defective immune response, this may lead to further accumulation of immune cells in the lungs, causing overproduction of pro-inflammatory cytokines. And this damages the lung structure. And the resulting cytokine storm that remains, I'm talking about the phase 3, it actually leads to multi-organ damage. That is actually, which is what we could see, the dysfunctional immune responses. But if we look at the healthy immune response, the initial inflammation attracts virus-specific T cells to the site of infection, where they can eliminate the infected cells before the virus spreads. And the neutralizing antibodies in these individuals can block the viral infection, and alveolar macrophages recognize neutralized viruses and apoptotic cells and clear them by phagocytosis. Altogether, these processes lead to clearance of the virus and minimal lung damage, resulting in recovery. That's what I'm saying, which is actually is reflected in the population where in general the population is always under the stress of different types of infection. Their immune system is pretty strong, and this healthy immune response in this case actually becomes very, very good to fight against the COVID. Whereas if you look at the European countries and specifically the United States, which are affected badly with this, their healthy immune responses are not there. And so the dysfunctional immune responses are more of the reason why we could see more death in the United States and huge number of infections every day. Even now, almost 0.2 million infections per day. Now, in the next slide, I just mentioned to you about the potential. Next slide. Next slide. This slide I already explained to you. Yeah. Potential therapeutic approaches against the SARS-CoV-2. So the first one is the antibodies to spike resulting from vaccination or adaptive transfer, which block virus binding to ACE2. And this could be done by the vaccination by the attenuated virus or the dead virus. And you know that the NIV, National Institute of Virology, they identified a SARS-CoV-2 strain, which is less infectious, a little bit mutant, but can produce the same response. So they actually grown that one because that cannot cause the infection and to this extent, and develop this attenuated virus vaccine, which is Covaxin. And this Covaxin is in the phase 3 trial right now. It was done by the Bharat Biotech. So the Indian scientist, NIV, ICM or Bharat Biotech jointly coming up with this Covaxin, which is under the phase 3 trial. Even in Calcutta, in NICET, they're actually giving this phase 3 trial is going on. And I'm fortunate to be a volunteer of that particular trial. And so I had that thing already on 4th. The second one is a protease inhibitor, which blocks and prevents the TMPRSS2, because if we block TMPRSS2, so it will not activate. And if it doesn't activate, then it cannot infect. The third one is the virus-specific effector, CD8 plus T cells, which arises from vaccination, recognize the infected cells, secreting cytotoxic granules to kill infected cells. And the fourth one is a pro-inflammatory cytokines, which can be neutralized through transfer of blocking antibodies through mechanical removal of it from the blood. Now, host response to SARS-CoV-2 in comparison to any other, next slide. Next slide. In comparison to other respiratory viruses, SARS-CoV-2 infection, it drives a lower antiviral transcriptional response that is marked by low interferon 1 and interferon 3 levels and elevated chemokine expression, which could explain the pro-inflammatory disease state associated with COVID-19. You can see that most of these references I'm using, these are actually coming out in the different top journals in only 2-3 months, from April and May, June, July, etc. And it was found that host cell, next slide, you can see the Nature publication, which shows the host cell translation changes upon SARS-CoV-2 infection. And if you look at the genes, which are, it starts from the base excision repair to the ribosomal RNA processing, and you can see the translation elongation, signaling by robo, MHC class 2 antigen, regulation of messenger RNA stability, Parkinson's disease, Huntington disease, nucleobase biosynthesis, all these particular type of things have been affected by this. And this is actually done by this transcriptome analysis with 2-hour, 6-hour, 10-hour, 24-hour pulse with 11-plex MS sample and followed by the LC-MS-MS. Next slide. It shows a pathological inflammation in patients with COVID-19. And you can see that the different types of monocyte-derived inflammatory macrophages are actually accommodated. And at the end, the cytokine storm, you can see that all this IL-6, TNF, IL-8, IL-10, CXCL10, IL-14A, IL-1RA, all these things are actually there. But in the meantime, you have the involvement of the activated T cells, NK cells, then we have the oxidative stress developed with the help of the TRAM-TRIF pathway and goes to the inflammatory genes through the NF-kappa B. And you can see that activated endothelium and the classical monocytes, they also play a very important role in this pathological inflammation. In the next slide, another aspect of this COVID-19 is the complement activation, which may contribute to the maladaptive inflammatory response seen in some patients with severe COVID-19. And inhibition of this complement C3 or C5 may have therapeutic potential, and people are also working on it. So at first it was thought remdesivir, which can affect virus replication, or ritonavir, lopinavir, but now the WHO already announced remdesivir is not doing such a huge work, though till now it's being used for actually treating the patients. And then you can see the activation of innate immunity by the pathway of specific inhibitors or anti-C3 or anti-C5, anti-IL-6, anti-IL-1 or anti-interferon. All these things people are looking at, and in general, it is actually, we could see that this, how after which particular period, if you add it, that can actually have an effect on the system. It may be from 1 to 2 days, 3 to 5 days, 5 to 7 days, and 7 to 14 days. In the next slide, the possible contribution of the hyperactivated monocytes to coagulation have been shown in the COVID-19. And I don't want to go through this slide because of the time, but I just wanted to mention that major endogenous anticoagulant pathways, which include the tissue factor pathway inhibitor TFP1, antithrombin, protein C, are reduced further, supporting the coagulation activation. And so in general, the heparin treatment has always been there, but thinners are there. In the next slide, I come to the main and the last point to be discussed, that COVID-19 discriminates age, gender, immune system fitness, pre-existing diseases, and all this may be correlated with our epigenetic factors. In general, the knowledge what we have from long time back, almost 2017, very nice paper came out, the respiratory viruses and histone modification, where infection with pathogenic influenza viruses and coronaviruses induces changes in the basal state of host chromatin. H1N1 and the SARS-CoV-2 results in enrichment of the H3K4-ME3 incorporation. and depletion of H3K27, and therefore in open transcription active chromatin. Whereas in contrast, H5N1 or the influenza virus VN1203 and the MERS, Middle East Respiratory Syndrome coronavirus, its infection drives H3K27 ME3 enrichment and depletes the H3K4 ME3 for a subset of genes, favoring a closed chromatin conformation. So one is the active chromatin conformation and another the closed chromatin conformation that inhibits the interferon-stimulated gene expression. Now the coronavirus-dependent host epigenome alteration, next slide. Next slide. Coronavirus-dependent host epigenome alterations and potential interventions are very interesting to find out from this particular slide if you look at. So host infected with the virus, the DNA methylation pattern, the histone modifications, and the non-coding RNA methylation, non-coding RNA or RNA methylations in siRNA, miRNA, gap RNAs, RNMT1, RNA methyltransferase inhibitors, all these things are being altered. The host epigenome alterations could be shown in all these cases. So from HTAG, SHARP, PPA, panobinosat, HKMT1, the DZ, NAP, HAT, anarchidic acid, IMG 149, all these things are being altered by this. So what we can see, the epigenetic machinery is responsible not only for the host response priming and memory, but also for ensuring its functional regulation. The age-related alterations to the host epigenome might affect the adaptive immune response, hindering the viral defenses. So epigenomics represent a powerful tool to explore how to prevent, attenuate, or reverse a viral infection therapeutically. The enzymes responsible for the epigenetic alterations might represent potential targets for new antiviral drugs. In the next slide, I just wanted to mention about this next slide, the COVID-19 fatality risk. Now, epigenetic dysregulation of the immune system and of the renin-angiotensin system may increase fatality risk. A variety of biological clocks have been shown to predict human health and longevity more accurately than chronological age. An individual with a biological age greater than their chronological age is thought to be undergoing accelerated aging, which may increase the risk of COVID-19 fatality. Individuals with comorbidities such as cardiovascular diseases, diabetes, obesity, and COPD are at greater risk for COVID-19 fatality. Conversely, individuals who live healthy lifestyles, consume geroprotectors such as metformin, resveratrol, and NAD boosters may have a decreased risk of fatality. Now, you know the diabetic patient, normally they have the metformin, and that's actually how it gets. So the epigenetic regulation, which gives you youthful immune cell composition, the biological age, chronological age, geroprotectors, healthy lifestyle, and if it dysregulated, the cytokine storm, the immunosenescence inflammaging, the biological age, chronological is always going to be greater than the chronological age. And the environmental factors, comorbidities present very, very important role. Next slide. I just wanted to show the epigenetic mechanisms in SARS-CoV-2 susceptibility. Now you can see that women, children, and non-smoker, the CpG island, that you can see this. Now, within the methylation pattern, it says the cells with less SE2 and interferon. So women, children, and non-smokers, you can see the less. And that actually protects against this COVID-19. But the man, elder people, or the smoker, you can see the CpG island. There are less methylation, cells with more SE2 and interferon, and so susceptibility is higher. So the expression of SE2 and interferon gene depends on the methylation rate of the CpG islands in the DNA promoter sequence. And susceptible individuals, mostly men, the elderly, the smokers, smokers of any gender shows a hypermethylation pattern of the ACE2 and interferon genes, whereas women, children, and non-smokers should get a hypermethylation and lower expression of ACE2. So that has got some relationship between this SARS-CoV-2 susceptibility. Next. And with the comparative analysis of COVID-19 case fatality rates by country, sex, and age being done, you can see it's very interesting result over there. This is from all the different countries, Australia, Iran, South Africa, to Netherlands, England, everywhere. In general, if you look at the female counterpart has got less numbers, but than the male counterpart. And in the case fatality ratio also, we could see that the male counterparts are actually always having a much higher level of effect. So if you look at the country, it's interesting to see the differences. You can see Bangladesh, Pakistan, Thailand, these Asian countries, Ukraine, even in China, you can see it's very controlled. But if you see from the England, Netherlands, Italy, Sweden, or United States. United States is not so high, but England is very high actually with respect to that. United States actually is getting more of this fatality. Now, known sex differences, next slide. Next slide. Known sex differences that may impact immune responses to SARS-CoV-2 and COVID-19 progression. This is actually would be identified with the PMPRS2, like the androgen regulated in prostate cells. Virus cells in and the antiviral response, you can see the TLR7, increased interferon gamma, or the PDC responses, or the hormone regulated. So the control of infection or resolution of inflammation and the progressive infection cytokine storm, all these two are actually being correlated in this particular slide. So to give you some idea about the SARS-CoV-2 and the COVID-19 progression. And the next slide, I will try to explain to you why does COVID-19 disproportionately affect the older people. Now, I tried to explain it, but if you look at the youthful system, the SARS-CoV-2 infection from the upper respiratory tract to the lower respiratory tract, to the immunosenescence, inflammaging, viral antagonism, ARDS, cytokine storm, coagulopathy, you can see the aged system, the suppressed immune system is actually the one. So the aged system, the viral alert signals are initially slow, resulting in a greater viral replication. The defective macrophages and T cells with a limited repertoire of receptors are less effective. More cells are infected, inducing high levels of inflammatory cytokine signaling. On the other hand, if you look at the cytokine storm, initiates a microvasculature clotting, causing severe hypoxia, coagulopathy, and organ failure. The CPGs in the ACE2 promoter decreases with age, and these CPGs are bounded by long-range promoter enhancer contacts that may change over time. And that's why we could see why COVID-19 disproportionately affect older people. But if for some reason or other, this happens to the younger generation also, it will also act. So for example, smoking, a big smoker, or having a lot of comorbidities, all these things. So the age, next slide. You can see this is actually to just show you the changes in T cell diversity, inflammaging, NAD plus loss, inflammasome activation, dysregulated renin-angiotensin system, changes to the glycome, advanced biological age, epigenetic changes, immunosenescence. All these are correlated with this COVID-19 susceptibility. So in general, what we could find, the glycome, which controls a variety of immune signaling pathways, it changes during aging and in the context of metabolic diseases. For example, decrease in IgG galactosylation contribute to chronic inflammation. Now, Biological clocks that measure the different biomarkers of biological age may explain increased COVID-19 susceptibility more accurately than advanced chronological age. And thereby what we can find that coronavirus also possesses an ADP ribosyl hydrolase that further depletes already low NAD+ levels in older people. So leveling of the epigenetic landscape during aging results in changes in immune cell composition and function that decreases the immune system's ability to mount a response to infection. In the next slide, we could see the different literature curation of genes associated with key COVID-19 morbidities. Now, the text mining approach to retrieve genes in abstracts associated with 6 human diseases has been shown in this slide. And the number of genes present in at least 4 abstracts of a disease is shown in the pie chart. The knowledge, so that is what we can see in this. In the section B, the knowledge-based network of COVID-19 morbidities, the network shows the disease, which are shown by the red nodes, and genes from panel A. The ages represent an association between disease and the gene. The size of the node is proportional to its degree, and genes associated in this part C, it's been shown that genes which are associated with 4 or more COVID-19 morbidities. So this is actually one which is a preprint right now, not yet published, and the most recent literature which came out. And you can see that there are almost 8,727 abstracts which shows about these genes and the number of genes which are associated with the different things which has been shown. So for example, the highest number we could see with hypertension, 392. The hypertension pulmonary is a blue part, 203. And the next highest one is the one which we call as a cardiovascular disease, 93. Autoimmune diseases, it's not so high. Similarly, renal insufficiency is also not so high, what we could find. So all these things could also be very nicely reflected in the section B, and in the section C, the genes are really mentioned over here. With this, I just wanted to say that next slide. Next slide. The epigenetics in COVID-19 immune syndrome and targeted therapies, which have been actually people are working out, and in one of the paper recently appeared in the clinical epigenetics, the main pro-inflammatory cytokines and the chemokines are upregulated by effector cells due to the hypermethylation acetylation. The ultimate goal of such intervention is to upregulate anti-inflammatory cytokines and to deplete pro-inflammatory cytokine levels through epigenetic modulation, which are going through this huge number of work which is coming up, and it very nicely shows that how the cytokine storm is being activated in this particular, is very strongly related with this epigenetic signing. In the next slide, the comparative, next slide is a very complicated slide. We are showing that comparative transcriptome analysis, which reveals the key epigenetic targets in SARS-CoV-2 expression. And here, it's also from another preprint, the differential expression analysis of coronavirus infected cells are actually been there. And it was found that the top 10 simplified enriched gene ontology in terms of biological processes and the virus associated gene sets ordered by the Q value. And this comparative transcriptome analysis is actually gives us an idea about the different related to the modification of RNA, the DNA modification, the histone writer cofactor, or the history writer. All these things are being very nicely, actually, right now we're coming out. And it's actually the functional classification of the AP factors whose expression is altered by SARS-CoV-2 infection has been shown in this slide. It's a very complicated slide. I don't want to say, but what I just want to mention that in this short period of time, a huge amount of work in the last 2 months, I could see a huge amount of papers coming out to correlate the epigenetic regulations and to find out actually the reason behind the differential effect of this coronavirus too. Next slide, the comparative transcriptome analysis. Next slide, the reveals the key epigenetic targets in SARS-CoV-2 infection. So over here you can see that nucleosome occupancy is one, histone modification is the other one, DNA methylation is another one, and the transcription factors. So all these four are being contributed with respect to this epigenetic targets in SARS-CoV-2 infection. So it's very interesting to find it out that these are actually being really affecting the system in a very differential manner amongst the population. You can see there are a lot of questions about the symptomatic and asymptomatic. Why there are so many asymptomatic? What are the reasons for the asymptomatic one? All these things could right now we could start understand with respect to this epigenetic signages. And next slide. The ACE2 regulation in the lung ventricle are organized, it was found that pathways enrichment analysis revealed that several of the genes positively associated with ACE2 were regulated by KDM5P and by specific histone acetylation H3K27AC and histone methylation, the H3K4ME1 and H3K4ME3. In the human lung, peaks for H3K4ME1 and H3K4ME3, as well as H3K27 acetylation, were identified in the ACE2 locus, suggesting ACE2 may be epigenetically regulated in the lungs. So if epigenetically regulated in the lungs, ACE2, so you can see the primary target is actually regulated, and epigenetic thus contributes in a very big way in this COVID-19. So in the next slide, the mechanisms that might increase susceptibility to and severity of COVID-19 lupus patients. So lupus patients show evidence of ACE2 hypomethylation, possibly mediating ACE2 overexpression, which will increase the likelihood of SARS-CoV-2 infection. It's very interesting that interferon-regulated genes and other inflammatory cytokine genes are hypomethylated in lupus patients and thereby epigenetically primed for transcription of an interferon exposure resulting from viral immune response. This epigenetic priming might increase the possibility of cytokine storm in the lupus patients, which came out in clinical immunology in the last month. In the next slide, I'm just going towards almost the end of my presentation. It shows the ACE2 expression profile that indicated 27 different tissues. Now, over here you can see duodenum, the gallbladder, the kidney, the small intestine, and the testis, where you can see that this is quite hard. In the others, it's not. So ACE2 expression profiling distributes were organ specific, mainly the kidney, male testes, female breast, and cardiovascular and gastrointestinal system. The next one is just to correlate with the cancer patients. Next one, next slide. ACE2 expression profiles in cancer patients. So the high expression of ACE2 was correlated with increased survival rate in renal and liver cancer, indicating that ACE2 is a prognostic marker in both renal cancer and liver cancer. Thus, ACE2 is a functional receptor for SARS-CoV-2 and has a potential anti-tumor role in cancer. So this is how we can correlate with the cancer patients. In the next slide, I just wanted to show you the RNA expression aberration of AC2 in tumors using GP1A2 and Wellcome database. And thereby it's been shown how these two are actually being changed. And the next slide, the colon adenocarcinoma, the kidney renal papillary cell carcinoma, pancreatic adenocarcinoma, rectum adenocarcinoma, lung adenocarcinoma, and stomach adenocarcinoma, which presented increased AC2 expression. And here the results are like this, which actually says that AC2 upregulation means what? AC2 upregulation means more infection. Next one. Next slide. I just want to say that testicular germ cell tumors, thyroid carcinoma, kidney chromophore presented the decreased AC2 expression. So in those cases, what we can see, how actually it's going to be affected by the COVID-19. So in the next slide, the two Wellcome and the promoter methylation level in AC2 in TG-SUD, it actually shows that high AC2 expression was associated with decreased DNA methylation level of AC2 in the different cancers. Whereas in the other one, I am showing you AC2 down-regulated tumor, testicular germ cell tumors, and the presented increased DNA methylation level. So these are actually how we can find it out. The next slide. Next slide. The major genetic risk factor for severe COVID-19 is inherited from Neanderthals. This is one of the most recent paper I've gone through, and thereby phylogenetically relating the DNA sequences that cover the core Neanderthal haplotype in individuals from the 1000 Genomes Project and Neanderthals. It actually shows this one. The colored area highlights a haplotype that carried the risk allele, that is the risk haplotype for severe COVID-19. The Arabic numbers indicate the bootstrapped support. The phylogeny is rooted with the inferred ancestral sequence of present-day humans. The three Neanderthal genomes carry no heterozygous position in this region. So this is very, very interesting information. And this actually will give us a new light into the research in the area of this. And in the next slide, I can show you the major distribution of genetic risk factor. Major genetic risk factor for severe COVID-19 is inherited from the Neanderthals. Now, the geographical distribution of the Neanderthal core haplotype that confer haplotype that confers risks for severe COVID-19. The pie charts are actually show the minor allele frequency at RS3 5044562. The frequency data were obtained from the 1000 Genome projects, and you can see that the area where you can see the population of Neanderthals is higher than any other places. With this, I just want to share the huge thanks to the health workers on the front line of the coronavirus pandemic. And my request to all of you, next slide, please. Next slide. Stay safe, stay home, stop coronavirus, and stay positive. And in that way, actually, we can still work out and defend ourselves from the coronavirus. Thank you very much. Thank you, everybody, for your helping me to deliver my understanding to this very, very complicated topic. Thank you. Namaskar.
Thank you, sir. Thank you so much, Professor Chattopadhyay. The most amazing part of any discourse that I attend of yours is that you take the whole thing from a personal, anecdotal point of view, like you discussed, like you told us about your interaction with Professor Archana Sharma, our beloved Didi. Regarding epigenetics, and bringing it all over to the current scenario and putting everything in perspective so wonderfully, and just hitting upon the point that the epigenetics can help us understand so much about what we really didn't understand even a few months ago about this, the correlation between all these morbidities and all the epigenetic modifications and sex, aging, and all of these. So actually the most beautiful part of your lecture, sir, I always feel is the question that comes to that mind. Immediately in a few slides, you really do answer that. So thank you so much. And I will ask the participants, if you have answers, if you have questions, I'm sorry, you can put it in the chat box. We already have one from Professor Shormishta Rajodhuri. Sir, may I present the question to you?
Yes, please.
Professor Rajodhuri is asking, does AC2 influence breast cancer patients?
Actually, it's very interesting that breast cancer patients, this patterning of ACE2 is not so much different. And that's why from the analysis, it's only find out the particular cancers which I mentioned about, that where the high ACE2 expression, which were associated with the decreased DNA methylation, it's for the colon cancer, for the kidney cancer, for the pancreatic cancer, and the rectum cancer. But in case of the testicular or in case of the other, it has been shown it's down-regulated. But in case of breast cancer, it's almost similar actually. There are no differences actually.
Professor Moshumi Poddar Sarkar asks that ACE2 is down-regulated in kidney chromophore, but the kidney patients are more affected by COVID-19. So how can we correlate that?
Yeah. So thereby I have shown you that if you remember the promoter methylation level, in case of this kidney papillary cell carcinoma, we have high ACE2 expression. Now it is the other way around when we looked at this in general, the kidney patients who are actually associated with this papillary cell carcinoma or with similar type of things that are high ACE2 expression. So that is not really true for the ACE2 down regulation. So thereby we could see, if you find out the association slide, which I have shown to you earlier, that how really this is highly associated between the two.
Thank you, sir. And I think you have also outlined the immense efforts that the scientific community has put up in the last few months and churned out such a huge amount of information that will take, years actually to make sense of. And I think now the next thing in our agenda is I will request Professor Anita Mukherjee to deliver the customary vote of thanks. Professor Mukherjee.
Thank you, Momita. But before that, we had as a token of appreciation, we would like to felicitate Professor Chattopadhyay. Are you ready with that?
Yes, yes, ma'am, I am ready with that.
I think you can do it now, and then I'll go for the
Okay, sure,
customary
sure.
vote.
Okay, I will. So since these are such unusual times, we have a photo of the memento that we want to present to Professor Chattopadhyay, and I will share of the image of that with you, sir, and with your permission, I will myself take it to you whenever you are available and hand it over. But at this point of time, this is our humble submission that we'll be very happy to share this. Can you all see this? So this is the plaque that would be presented from the Archana Sharma Foundation of Calcutta to Professor Dhrubajyoti Chattopadhyay for the oration lecture.
I gladly accept it, and thank you, Momita, that whenever we will meet, I can get it.
Thank you so much, sir. So now I think Anita ma'am, Professor Mukherjee, you can take over.
So On behalf of the Archana Sharma Foundation of Calcutta, it's a privilege for me to offer this vote of thanks today. I extend my sincere thanks to our Honorable Speaker, Professor Drubojyoti Chattopadhyay, Vice Chancellor of Sister Nivedita University, known to us as Dhruboda or DJC, to take time out of his busy schedule and do this honor today. Thank you very much for the nice and lucid presentation, which was not only informative as well as very much updated on today's. And we had the idea about the background, why such a, like Momita has already summarized, so I won't go into details, but I was also mesmerized by the lecture to know the facts also, how why is this COVID-19, this one is so different from the other viruses, why it spreads so fast, and what is the background of such unique sequences, responses which you have explained in terms of epigenetics. Thank you very much, Dhruboda.
Thank you.
And
I just
we
want
would
to
also
thank, I could see Professor Lavania's also reaction. I thank Professor Lavania and everybody, all my friends who are present today. And I really give my thanks to the foundation for giving me this opportunity.
Now, we would also like to thank...
I enjoyed the lecture.
We would like to thank also the head of the Department of Botany, University of Calcutta, Dr. Binay Chaube, and his team for his support and help in organizing this oration. Now, without the participants, our endeavor would not have been materialized.
It is always a pleasure to listen to sir.
Yeah. But but the foundation also acknowledges your support definitely, and also the department's help. And without the participants, our endeavor would not have materialized. So we thank all members of the foundation. As I can see, our senior member, Omyang Shuda, is here, as well as Tondra, other teachers from nearby colleges, Shushobun Bera and others. Don't mind if I miss the names. And I also would like to thank all the ex-scholars and student participants who had helped us and has joined and made this event successful. Lastly, but not the least, I would like to thank Mr. Akash Mondal of Brain Drops for his technical support. And thank you all, and I wish you a very Merry Christmas and a Happy New Year ahead. Thank you.
Yes. Thank you very much.
Just a small announcement. There is a feedback form that has been shared in the chat box. We will be very happy if you please leave your comments in the form that has been shared with you. That is just for our our satisfaction that the whole event, if there was some problem technically with the event, we really regret it. Even we are getting adapted to the changed circumstances, the neo or the new normal, and probably all the epigenetic resetting is not complete. So there are a few still obstacles probably, but thank you all so much. Thank you, Professor Chattopadhyay, so very much for this wonderful discourse. And thank you to all the people who have joined us today for making this. Yes, sir.
When you said that there is a small announcement, I thought that you are going to announce that there is a tea outside.
So this is the new normal. Yeah, this is the new normal, Professor Chaube. We are not yet epigenetically reset for that.
Yes.
We are still expecting our tea and a small chit chat over the tea at the end of a wonderful discourse like this. But yes, we will come back to the actual original normal that we are all used to. So regretting the new normal and the modifications we have to adapt to for that. Thank you all so much. For making this event a success. So on behalf of the Archana Sharma Foundation, I really sincerely appreciate all your help and endeavor and patience also. And thank you.
Thank you very much.
Thank you.
Thank you.
Thank you.
Thanks to everybody. Yeah.
Thank you, sir.
Thank you, sir.
Manoj, yeah, nice to see you. Yeah,
yes, sir. Excellent lecture, sir.
Okay. Thank you.
Okay, sir.
Thank you. Thank you, sir.
Thank you.
You can see Shondip Poddar also. Wow. He's joined
হ্যাঁ
from
দিদি
Malaysia.
হ্যাঁ
Kuala Lumpur থেকে জয়েন করলে? বাহ, ভেরি গুড।
এটা রিউনিয়নও হয়ে গেল। সন্দীপ দা ভালো আছেন?
হ্যাঁ অহিমাংশুদা কেমন আছেন?
হ্যাঁ,
মিউট রয়েছেন আপনি, বাণীদি রয়েছেন, তন্দ্রাদি রয়েছেন,
হ্যাঁ,
অহিমাংশুদা মিউট রয়েছেন, শোনা যাচ্ছে না।
না শুনতে পাচ্ছি না।
হ্যাঁ
আনমিউট করুন স্যার, আপনাকে দেখি একটু অনেকদিন বাদে। কেমন আছেন? আনমিউট করুন।
মৌমিতা, তুমি আমাদের এই ইউটিউবের লিংকটা পাঠিয়ে দিও, অনেকে যারা এখানে প্রেজেন্ট থাকতে পারেনি তারা চেয়েছে।
হ্যাঁ ম্যাম, দিচ্ছি এক্ষুনি।
হ্যাঁ, আমি ওদের পাঠিয়ে দিচ্ছি।